Top carnivorous plants, plants that eat animals. Part 1. Did you know that some plants can actually capture and digest insects? From cunning traps to ingenious adaptations, these plants have evolved some truly remarkable ways to secure their next meal. So, if you're ready to embark on a journey through the plant kingdom's dark side, keep watching. You won't believe what these botanical predators are capable of. But first, don't forget to leave a like on the video, subscribe to the channel, and click the notification bell to stay updated on our upcoming videos. Are you ready? Let's begin! 20. Venus Flytrap. The Venus Flytrap, Dionaea muscipula, is a carnivorous plant native to subtropical wetlands on the east coast of the United States. Its unique trapping mechanism is formed by the terminal portion of its leaves, triggered by tiny hairs called trigger hairs, or sensitive hairs, on their inner surfaces. This plant thrives in nitrogen and phosphorus-poor environments like bogs, wet savannas, and canebrakes. It's often found in areas with less than 10% canopy cover. While it's been successfully transplanted in various places worldwide, it's native only to the coastal bogs within about a 100-kilometer radius of Wilmington, North Carolina. The Venus flytrap primarily preys on insects and arachnids. It has a remarkable ability to distinguish between living prey and non-prey stimuli, such as falling raindrops. To trigger its trapping mechanism, 2 stimulations of the trigger hairs are required within about 20 seconds. The trap's rapid snapping shut is a result of complex interactions involving elasticity, turgor pressure, and growth. The trigger hairs play a crucial role in this mechanism, ensuring the trap doesn't close inadvertently due to wind-borne debris. The edges of the lobes are fringed with stiff cilia, preventing large prey from escaping. Once the trap seals, it forms a stomach, where digestion occurs. Digestive enzymes, including GH18 chitinase, break down the prey's chitin-containing exoskeleton. Oxidative protein modification and the presence of redox cofactors may also play a role in rendering the prey more susceptible to proteolytic attacks. Digestion typically takes about 10 days, after which the trap reopens, ready for reuse. 19. Cobra Lily. Darlingtonia californica, commonly known as the California pitcher plant, Cobra Lily, or cobra plant, is a unique carnivorous plant. It represents the sole member of the Darlingtonia genus within the Droseraceae family. This intriguing plant is native to Northern California and Oregon, USA, and thrives in cold, running water environments found in bogs and seeps, typically on serpentine soils. Its name, Cobra Lily, originates from the striking resemblance of its tubular leaves to a rearing cobra, complete with forked leaves that mimic fangs or a serpent's tongue. Cobra lilies are not confined to nutrient-poor acidic bogs and seepage slopes. They also thrive in ultramafic soils within their range. These soils, although toxic to most plants, pose no challenge to cobra lilies. They often grow in regions with cold subsurface water slowly flowing around their roots, which contributes to their unique adaptation to such environments. Like many carnivorous plants, cobra lilies have evolved to supplement their nitrogen requirements through carnivory due to the scarcity of available nitrogen in their habitats. They capture a variety of prey to compensate for this deficiency. Unlike other American pitcher plants, Darlingtonia californica does not trap rainwater in its pitcher. Instead, it physiologically regulates the water level within the trap, maintaining a balance by releasing or absorbing water pumped up from its roots. Its slippery pitcher walls and hairs prevent trapped prey from escaping. The plant also employs a curled operculum, hood, to obscure the exit hole from trapped insects, offering multiple translucent false exits to exhaust prey and lead them into the trap. While it was previously believed that cobra lilies relied solely on symbiotic bacteria and protozoa to digest captured insects, recent studies have shown that they do produce at least one proteolytic enzyme to break down prey. The plant's leaves and pitchers are often teeming with insects and their remains, showcasing their efficient trapping ability. Cobra lilies remain a challenge to cultivate, requiring specific environmental conditions, including cool to warm temperatures during the day and cold nights. They are best propagated from stolons, which can be cut into sections for growth. Like other temperate carnivorous plants, they undergo a cold winter dormancy period. Cultivators have also identified different color morphs, including all green, all red, and red-green bicolor variations. 18. Sundews. Drosera, commonly known as sundews, belong to one of the largest genera of carnivorous plants, comprising at least 194 species. These plants, part of the Droseraceae family, employ stalked mucilaginous glands on their leaf surfaces to lure, capture, and digest insects. This carnivorous behavior compensates for the poor mineral nutrition of their soil. Sundews exhibit significant variation in size and form and are found on every continent except Antarctica. Sundews inhabit diverse ecosystems, from bogs and marshes to rainforests and deserts. They favor acidic soils with ample sunlight. The genus has a worldwide distribution, ranging from Alaska to New Zealand, while Australia, South America, and Southern Africa host the greatest diversity. Sundews are also found in Eurasia and North America, though not in temperate or Arctic regions. Sundews predominantly prey on small insects, attracted to the sweet mucilage secreted by stalked glands on their leaves. This mucilage entangles and ultimately suffocates the trapped insects. Sundews exhibit thigmonasty, rapidly bending their sensitive tentacles towards prey upon contact. Some species can also bend their leaves to maximize contact with the trapped prey. The trapping and digestion mechanism of sundews involve two types of glands: stalked glands for ensnaring insects and sessile glands for absorbing nutrients. Sundews secrete enzymes such as esterase, peroxidase, phosphatase, and protease to dissolve and extract nutrients from captured insects. Death typically occurs within 15 minutes of entrapment. Upon capturing insects, sundews release enzymes that break down the prey's tissues, releasing nutrients. These nutrients are then absorbed through the leaf surfaces to nourish the plant. Sundews play a vital role in their ecosystems, adapting to various environments and exhibiting remarkable carnivorous adaptations. They face threats such as habitat destruction through urbanization and agricultural development, illegal collection for horticulture, and changing environmental conditions. Conservation efforts are crucial to protect sundews from extinction, particularly in regions where their survival is most endangered. Which carnivorous plant do you think has the most elaborate and intricate trapping mechanism? Tell us in the comments. 17. Aldrovanda vesiculosa. Aldrovanda vesiculosa, commonly known as the water wheel plant, is a unique flowering plant belonging to the Droseraceae family. It is the sole surviving species within the Aldrovanda genus. This remarkable plant employs carnivorous traps akin to those of the Venus Flytrap and exhibits rapid movement, making it one of the few plants capable of such motion. Aldrovanda vesiculosa is a rootless aquatic plant that thrives in clean, shallow, and warm standing water. It requires bright light, low nutrient levels, and a slightly acidic pH for optimal growth. The plant's habitat spans across Europe, Asia, Africa, and Australia. Its distribution is mainly facilitated by waterfowl, which transport Aldrovanda to new aquatic destinations as the plants adhere to their feed during migration. This carnivorous plant displays a selective appetite for small aquatic invertebrates. Its traps are finely tuned to capture suitable prey efficiently, with a preference for specific aquatic organisms. Aldrovanda's trapping mechanism is ingenious. Its traps consist of two lobes that snap together underwater when triggered by contact with aquatic invertebrates. The traps are configured to prevent false triggers by debris in the water. This rapid closure, taking only 10 to 20 milliseconds, is one of the swiftest movements observed in the plant kingdom. However, this mechanism operates optimally in warm conditions of at least 20 degrees Celsius (68 degrees Fahrenheit). Once prey is trapped, Aldrovanda vesiculosa employs its carnivorous adaptations for digestion. The plant extracts nutrients from the captured invertebrates, aiding its growth and survival in nutrient-poor aquatic environments. Aldrovanda vesiculosa's fascinating characteristics and its role as a carnivorous plant contribute to its ecological importance and continued study among botanists and plant enthusiasts. 16. Nepenthes. Nepenthes, commonly known as tropical pitcher plants or monkey cups, belong to the monotypic family Nepenthaceae. This genus comprises around 170 species, both natural and cultivated hybrids. These liana-forming plants primarily inhabit the Old World tropics, spanning from South China, Indonesia, Malaysia, and the Philippines to Madagascar, the Seychelles, Australia, New Caledonia, India, and Sri Lanka. The greatest diversity of Nepenthes occurs in regions like Borneo, Sumatra, and the Philippines. Many species are endemic to these hot, humid lowland areas, while others thrive in tropical montane environments with warm days and cool nights. Some even adapt to cooler, near-freezing tropical alpine conditions. The name, monkey cups, originated from the mistaken belief that monkeys drank rainwater from the pitchers. Nepenthes species predominantly capture insects. Occasionally trap small vertebrates such as frogs, birds, and small mammals. Even instances of cultivated plants trapping small birds have been recorded. Each Nepenthes pitcher is filled with a fluid produced by the plant. This fluid, often viscoelastic, serves as a trap to drown prey. The lower part of the trap has nutrient-absorbing glands, while the upper inside part features a slippery, waxy coating called the peristome. The peristome's slippery surface causes insects to slip and fall into the pitcher. An operculum lid keeps rainwater from diluting the digestive fluid, and nectar glands above the peristome attract prey. Nepenthes digestive fluids are sterile before the pitchers open, containing antimicrobial proteins and secondary metabolites. These substances kill or inhibit the growth of bacteria once the fluid is exposed to the external environment. This adaptation helps the plant maximize nutrient uptake from captured prey. These complex ecological interactions with their environments, including predator-prey relationships and mutualistic associations, make Nepenthes unique and fascinating carnivorous plants. 15. Trumpet pitchers. Sarracenia, often called trumpet pitchers, is a genus comprising 8 to 11 species of North American carnivorous plants. These unique plants are part of the family Sarraceniaceae, along with the closely related genera Darlingtonia and Helianthera. Indigenous to various regions, they have evolved remarkable features to thrive in their environments. The distinguishing feature of Sarracenia is their funnel or pitcher-shaped leaves, specifically adapted for trapping insects. They range in size and structure but share common characteristics in their predatory mechanism. Sarracenia species are primarily found along the eastern seaboard of the United States, in Texas, the Great Lakes area, and southeastern Canada. Most species are concentrated in the southeast United States, except for Sarracenia purpurea, which can endure colder temperatures. These plants inhabit specific ecosystems such as fens, herb bogs, and seasonally wet grasslands. These habitats are typically acidic, nutrient-poor, and characterized by sand and sphagnum moss. Sarracenia prefer abundant sunlight with minimal shade. Sarracenia lures insects primarily through a combination of color, scent, and the secretion of nectar. These features attract prey to their perilous trap openings, especially the highly specialized peristome region. Various species exhibit adaptations that make it challenging for insects to escape. Sarracenia relies on static traps with no moving parts. Insects are enticed to the trap openings but encounter slippery surfaces that cause them to fall inside. As they descend, downward-pointing hairs and digestive glands make escape impossible. The digestive fluids in the pitcher break down the prey, while exoskeletons are typically left undigested. Inside the pitcher tube, distinct zones can be identified. Zone 1, the operculum, covers part of the pitcher and prevents excess rainwater from diluting the digestive fluids. It also guides prey toward the entrance. Zone 2, the peristome and trap entrance, produces nectar and features a treacherous surface that facilitates prey capture. Zone 3, located below Zone 2, has a surface with no footing and digestive glands. Zone 4, the final zone in most species, is filled with digestive fluids and more digestive glands. Zone 5, present only in Sarracenia purpurea, lacks glands and its function is unknown. Sarracenia faces significant threats in the wild, including urban development, habitat drainage for forestry, herbicide runoff from agriculture, fire suppression, cut pitcher trade for floristry, and plant trade. Efforts to protect Sarracenia include various state and federal legislation and conservation initiatives by organizations dedicated to preserving these unique plants. These fascinating carnivorous plants are easy to cultivate, and they play a crucial role in maintaining their fragile ecosystems, showcasing the delicate balance between predator and prey in the natural world. Have you ever witnessed a carnivorous plant catching its prey in action? Share your story in the comments. If you like this video, don't forget to leave a like and subscribe to the channel please. 14. Byblis. Byblis is a small genus of carnivorous plants, often referred to as rainbow plants, due to their captivating, mucilage-covered leaves that glisten in bright sunlight. Native to Australia and New Guinea, Byblis is the sole genus in the family Byblidaceae. It was first described by botanist Richard Anthony Salisbury in 1808, and there are currently 8 recognized species. Byblis species closely resemble Drosera, Sundews, and Droseraceae but can be distinguished by their zygomorphic flowers featuring 5 curved stamens to one side of the pistil. Unlike these similar-looking genera, modern classifications place Byblis in the Lamiales, while Sundews and Droseraceae belong to the Caryophyllales. They can be categorized into 2 groups, the Byblis liniflora complex and the Byblis gigantea complex. These plants are found in bogs, marshes, and seasonally wet sandy soils, typically in partial or direct sunlight. Byblis plants feature round, highly elongated leaves covered in glandular hairs that secrete mucilage to attract small insects. When insects touch the sticky secretions, they become ensnared. Byblis cannot move their tentacles or leaves to aid trapping or digestion. The mucilage-coated leaves capture insects, and unless the prey can escape, they either die of exhaustion or asphyxiate as the mucilage envelopes them and clogs their spiracles. Additionally, Byblis leaves have sessile glands responsible for secreting digestive juices. 13. Butterworts. Pinguicula, commonly known as butterworts, belong to the carnivorous flowering plant family Lentibulariaceae. These fascinating plants use sticky, glandular leaves to attract, capture, and digest insects, compensating for their poor mineral nutrition from the environment. With around 80 known species, they exhibit diverse characteristics across regions. Most butterworts are perennial plants, with only a few exceptions like Pinguicula sharpii, Pinguicula takaki, Pinguicula crenitiloba, and Pinguicula pumila, which are annuals. They typically form stemless rosettes. Butterworts thrive in various climates, leading to 2 main groups based on their growth conditions. While genetic studies don't necessarily support these groupings, they are convenient for horticultural purposes. Tropical butterworts, found in regions with seasonal water scarcity and low nitrogen resources due to acidic soil conditions. They either maintain carnivorous leaves year-round or form compact winter rosettes during dry periods. Temperate butterworts, native to cold winter climate zones, these species create winter-resting buds, hibernacula, during their dormancy period. Many butterworts alternate between carnivorous and non-carnivorous rosettes as seasons change. Butterworts are passive accumulators of small insects, and those with large wings that can fly. They can also derive nourishment from pollen and other plant parts that get trapped on their leaves. Their colorful leaves attract prey, increasing the chances of capture. Butterworts use sticky mucilage secreted by glands on their leaf surfaces to lure and capture prey. 2 types of glands are involved. Peduncular glands. These produce mucilage droplets, luring insects in search of water. The droplets also entrap insects, and upon contact, more mucilage is released. Sessile glands. Located on the leaf surface, these glands release enzymes for digestion when nitrogen from prey is present. The enzymes released by sessile glands, including amylase, esterase, phosphatase, protease, and ribonuclease, break down the insect's digestible components. The fluids are absorbed through cuticular holes on the leaf surface. Unlike some carnivorous plants, butterworts do not appear to use jasminates to regulate enzyme production. Alpha-amylase, a unique enzyme in butterwort digestion, suggests a distinct genetic adaptation for carnivory. 12. Brocchinia reducta. Brocchinia reducta is among the fascinating carnivorous members of the bromeliad family. What sets Brocchinia reducta apart is its ability to thrive in nutrient-poor soils, adapting to various environments by anchoring itself to rocks using its roots. Similar to other bromeliads, Brocchinia reducta forms water-storing cups through tightly overlapping leaves. These leaves are adorned with loose, waxy scales that have a remarkable ability to reflect ultraviolet UV light. This UV reflection serves as an efficient lure for many insects, as they are naturally drawn to UV light, which is also reflected by numerous flowers. Additionally, the water within the cup emits a sweet odor, further attracting ants and other insects. Native to Venezuela, Guyana, and northern Brazil, Brocchinia reducta thrives in soil with low nutrient content. It is most commonly found growing on the tepuis of the Grand Sabana Plateau, sharing this unique habitat with other carnivorous plant species. The smooth and waxy texture of Brocchinia reducta's leaves provides an unstable foothold for landing insects, causing them to slip and fall into the water-filled urns. Once trapped, the prey find it challenging to escape due to the slippery surface, leading to their eventual drowning. While there was initial debate about whether Brocchinia reducta could be classified as a true carnivorous plant, recent findings in 2005 demonstrated its carnivorous nature. The plant produces phosphatase enzymes, supporting its classification as a carnivorous plant. Enzymes and bacteria present in the water within the urns aid in the digestion of trapped insects, releasing nutrients that are subsequently absorbed by the plant's leaves. Brocchinia reducta's remarkable adaptation to nutrient-poor environments and its carnivorous strategy for acquiring nutrients make it a captivating and unique member of the plant kingdom. How important do you think these plants are in their ecosystems? What role do they play? Tell us in the comments. 11. Roridula. Roridula, derived from the Latin word roridus, meaning dewy, comprises a unique genus of evergreen insect-trapping shrubs. This solitary genus belongs to the Roridulaceae family and consists of 2 species. These shrubs have slender, woody stems that initially appear brown but turn gray with maturity. Their branches are sparingly branched and upright. The lance to all-shaped leaves are densely crowded at the tips of the stems. Roridula is endemic to the mountainous regions of the Western Cape Province in South Africa. Roridula dentata is found in Tulbagh, Clan William, and Ceres Mountains, while Roridula gorgonias occurs between Somerset West and Swellendam. These shrubs thrive in acidic, nutrient-poor, sandy soils, particularly in relatively moist areas, at altitudes ranging from 900 to 1200 meters for Roridula dentata and 150 to 925 meters for Roridula gorgonias. They primarily grow in humid locations, sharing their habitat with various Drosera species. Roridula, like Drosera, captures a diverse range of flying insects. Roridula gorgonias has an affinity for UV absorption, which attracts flying insects. Unlike Drosera, Roridula gorgonias secretes a highly sticky resinous substance, making it effective in capturing insects, even during rainy weather. Rather than bending tentacles towards struggling prey, Roridula possesses 3 types of glandular hairs, differing in size. Long tentacles are less sticky, allowing insects to come into contact with much stickier medium and short glands that immobilize the prey. Roridula's trapping mechanism relies on the adhesive nature of its glandular hairs. These hairs immobilize insects, but Roridula does not secrete enzymes to break down proteins like some other carnivorous plants. Instead, it houses bugs of the genus Pamaridia roridulae, which feed on the trapped insects. These bugs then deposit their feces on the leaves, providing nutrients to the plant. Pamaridia bugs have a thick, greasy layer that prevents direct contact between the resin on Roridula tentacles and the insect's cuticle, allowing them to move freely across the plant. Your support and curiosity drive us to keep exploring the diverse and intriguing world of nature. If you have any more questions or topics you'd like us to cover in future videos, please feel free to leave them in the comments section below. Your feedback is invaluable to us. See you in the next video.